In this tutorial, we will discuss how to import microarray files, use the annotation management tool, add experimental design information, and configure the analysis settings. IBD can import files from different technologies, but for the purpose of these tutorials, we will focus on Affymetrix WT arrays. For the Affymetrix platform, we need to import the cell files. For Illumina, raw microarray data can be exported from Bead Studio and Genome Studio in the form of text files. Plain ASCII tab delimited text can be imported for 2-channel data from platforms such as Agilent. Please refer to pages 25 to 37 in our user manual to get further information about the input files from other platforms. There are 2 modes of analysis workflows, a predefined gene expression guided analysis and a user-driven workflow. We will choose the predefined gene expression guided analysis. Once the input cell files have been selected, we need to configure our annotation settings to ensure that we have all the library files necessary for analyzing this data. We can check this button here to ensure that all the relevant library files are present, and we can update these files if necessary. We can further configure these settings by going to Annotation Management and installing the relevant files from our local files or from the manufacturer's website by clicking on Connection Settings. Using the Annotation Management tool, we can also update UniProt and Go data as well as gene sets, pathways, and upload any custom annotations. We are now ready to define our experimental design. Each of the input files need to be assigned to one of these groups for each of the different factors. In order to assign a file to a factor, we can simply select and drag the files into their relevant groups. Alternatively, we can use the Add button after selecting the files and the destination factor. The format of this file with the experimental settings should be a simple tabular text format containing the cell file name, the sample name, and the group to which it will be assigned. If you have more than 2 groups, then we can easily add new groups of factors. Additionally, we can rename existing ones as well as delete any group of factors that we do not require. Once this assignment is done, you may save it in a file if you wish to avoid having to repeat the same steps in the future for these samples if they're reanalyzed. We can also configure our analysis settings that you may want to specify at the start of your click-and-go analysis. These include the summarization procedures, the normalization strategies, as well as different methods for hierarchical clustering if you prefer not to use the default methods. Finally, check the Click and Go button to analyze your data based on the settings you have just specified. For further information and tutorials, please visit www.integromics.com.